Hello everyone, how are you today? Welcome to the YouTube channel Plantastic. Today we are going to look into the somatic embryogenesis that often play as an alternative to the micropropagation. Embryogenesis is a specialized mode of development by which a fertilized egg through a series of predetermined patterns of cell divisions and differentiation to form an embryo. It is the precursor of the next generation. The model plan to study the somatic embryogenesis is a carrot, which is scientifically known as the Daucus carota. Alright, somatic embryogenesis, which is known as SE, is the formation of embryos essentially through somatic cells. It occurs naturally as on the leaves of calycol. Plants need to differentiate to form the stem cell. Unless it is already meristematic, then it will develop through embryological stages to become a new plant. The induction of embryogenesis, embryo development, embryo maturation, and their conversion on germination to form complete plants is included in this somatic embryogenesis. Alright, let's look into the differences between the somatic embryogenesis and organogenesis. In somatic embryogenesis, it is bipolar, whereby there is the formation of root and shoot at the same time. However, in organogenesis, it is unipolar, which means that the regenerant in organogenesis forms only shoot or it forms only root at a time. Usually it's the two steps whereby it is stimulated to form the shoot first, followed by the root, or vice versa. Somatic embryogenesis needs to proceed through a single hormonal signal, whereby the organogenesis proceeds through two hormonal signals to induce shoot and then induce the root separately. For example, in micropropagation, we usually use the cytokinin to induce a shoot, followed by the oocyte to induce the root. Somatic embryogenesis is an artificial process in which a plant or embryo is derived from a single somatic cell. Organogenesis is the series of organized integrated processes in which it transforms an amorphous mass of cells into a complete organ in the developing embryo. Somatic embryogenesis is an artificial process that occurs only under laboratory conditions. Organogenesis is a natural process which occurs in nature and it also can be induced artificially. Somatic embryogenesis is used to induce somatic tissue, which is known as the embryogenic callus, and then it will develop into the somatic embryos. However, organogenesis involves the induction of vegetative tissues to form the organs. Lastly, somatic embryogenesis can be detected by no vascular connection between the regenerant and the maternal callus. For organogenesis, there will be the vascular connection between the regenerant and the maternal tissue, in which you will see that there is a connection of xylem and phloem between the organs induced like shoot or root from the maternal tissue. There are two types of somatic embryogenesis. First, indirect somatic embryogenesis, in which the S-plant initially undergoes rapid cell division to form a relatively disorganized mass of cells called callus. Somatic embryos then arise from the callus tissue. The second type of somatic embryogenesis is the direct somatic embryogenesis. It is the embryos formed directly from the S-plant without an intervening callus phase. So, the summary of these slides is, when there is no callus involved throughout the experiment, it is indicated as the direct somatic embryogenesis, whereas if there is any formation of callus, it is known as indirect somatic embryogenesis. There are several factors affecting the somatic embryogenesis. First, the selection and manipulation of the S-plant, which is the plant tissue. This is because the regeneration ability of each S-plant is vary with each other. For example, leaf might be the better S-plant as compared to shoot. Secondly, nutrient medium, as followed by the growth regulator, also the physical cultural environment. Next, the S-plant. The genotype dependent with significantly variation in the response between the cultivars. This means that, for example, seeds, which known as the immature zygotic embryos, is often show the great significance as compared to the monocots like cereals and palms and many dicots. Commonly, the S-plant used in the somatic embryogenesis will be hypocotyl segment, young leaves, embryonic shoot tip, and also young floral part. For the seed, which is the immature zygotic embryos, it possesses embryogenic competence, which is known as pre-embryogenic determined cells. More differentiated S plants have the cells that must be inducted to become embryogenic, and then it will be termed as the induced embryogenic determined cells. As for the genotype, somatic embryogenesis is a genetically controlled threat. Genotypic variation could be due to the varying endogenous levels of the growth regulators, which means that there will be somal clonal variation due to the supplementation of different plant growth regulators. Besides that, the medium and its composition is the great influence to induce the somatic embryogenesis. It was found that 70% of the successful cases of somatic embryogenesis is obtained from MS basal medium or its modifications. Whites or SH basal medium has been used with suitable supplements. He also found that the carbon source, which is sucrose, glucose, and galactose or lactose, are playing a major role in somatic embryogenesis, especially glucose and lactose or galactose, which are commonly used to induce the somatic embryogenesis in the study. For the nutrient components, nitrogen is the major factor in the induction of somatic embryogenesis. The source of nitrogen can be nitrates or other nitrogen sources such as casein hydroxylase and amino acids. Nitrogen is one of the requirements during somatic embryogenesis, which may be correlated to continue the synthesis of protein, nucleic acid, and reserved substances, as well as to maintain the appropriate pH. In somatic embryogenesis, general practice is to supplement oosin for callusing, which is 2,4-D that induce the callus to differentiate into the localized group of meristematic cells called as pro-embryogenic masses. Other oosin are also has been shown to promote the somatic embryogenesis, for example, dicamba for monocots, IAA and NAA. On the other hand, cytokinins like TDZ and zeatin also being used in the induction of somatic embryogenesis. Other plant growth regulators are also being found to have the effect of induced somatic embryogenesis, which are gibberellin, parasinolite, high sucrose concentration, and manipulation of pH. However, glucose and galactose or lactose are not commonly used in somatic embryogenesis study. Subsequently, we need to do the selective subculture, whereby the multicellular explants are generally a mixture of cells differing in their potential to form somatic embryos. However, from a big portion of callus, only small proportion of these cells are able to express their cellular totipotency under a set of culture conditions. Therefore, we need to pick up the potential cells and subculture it, and then proliferate it. The embryogenic portions of a callus may distinct from the non-embryogenic tissue on the basis of their morphological appearance. It can be granular and with smooth surface for some Poaceae species, which is grass species. But for coffee, which is known as a Coffea arabica, it will show brown, which is the phenolized and hard texture. In some study, electrical stimulation is found to be useful by applying the mild electric current, which is known to stimulate the shoot and embryo differentiation in somatic embryogenesis. It will induce the changes in the organization of microtubules, which affect the cell polarity. There are some other factors that will influence the regeneration activity, in which it could be the initial cell density, whereby the high cell density will inhibit the formation of somatic embryogenesis, which could be overcome by the addition of activated charcoal. This is because as the cells release a high amount of phenolic compounds like phenylacetic acid, benzoic acid, 4-hydroxybenzoic alcohol, PHPA has the strongest inhibitory effect on somatic embryogenesis. Therefore, it is better to incubate the cell at lower cell density. Other factors could be the ethylene, which is the gaseous phytohormone that accumulated in the vessel. Silver nitrate is able to increase the somatic embryogenesis in carrot and rubber. They suppress the production of ethylene. It is found that oxygen concentration plays a major role in somatic embryogenesis too. This is because the reduced dissolved oxygen in wheat cultures improve the somatic embryogenesis by almost 6-fold. The extracts of the prokaryotic origin, which is known as the blue-green algae extract, can be added to induce the somatic embryogenesis. You must take note that the light, in terms of the quality and photoperiod, is also important to influence the somatic embryogenesis as well as the application of some heavy metals. There are several conditioning factors regulating somatic embryogenesis at physiological level, which are extracellular proteins, arabinogalactan proteins, and lipocyto-oligosaccharides. Here is a general process of the somatic embryogenesis, whereby you can see the development of the somatic embryogenesis. For the induction of somatic embryogenesis, oocyte acts as the main PGR for dedifferentiation, which is 2,4-D. The polarity is the alternation of the cell polarity that promotes asymmetric division. The differential cell fate is determined by the physiological asymmetries with respect to unequal distribution of molecules like arabinogalactans and other constituents. It was found that electrical stimulation promotes the differentiation of organized structures like shoots or embryos by affecting the cell polarities, which leads to the changes in the microtubule organization. The development of somatic embryogenesis is pretty simple. Apart from the pro-embryogenic methods, which is known as the PEM, there is two types of single cell, which are cytoplasmically rich small cell that give rise to PEM, but not most of the PEM are derived from the pre-existing PEM. The second type of the cell will be the vacuolated long cell. The continued presence of oocyte even after the induction of somatic embryogenesis causes the cell elongation and disruption of the formerly adhering cells. Therefore, you will see some enlarged cells that permanently lose the competence of developing into somatic embryos. The embryogenic cells secrete certain proteins, which are used to maintain the embryogenic potential of the cultures by restricting the cell elongation in the presence of 2,4-D, and also to induce the appearance of small embryogenic cells in non-embryogenic cultures. Moreover, the disruption of the cells from each other stops, and the globulus develop into globular embryos when the embryogenic cultures are transferred to oocyte-free medium. The globular embryos then continue further development to form the typical embryos. ABA-abscisic acid is used to stimulate the epidermis development. The developing embryos synthesize their own oocyte and further morphogenesis is carried out that beyond the globular stage, which is dependent on the proper polar transport of the oocyte. And then, the somatic embryos will form on the surface of the callus, which have the intrinsic polarity. The pro-embryogenic methods will develop into the complete embryos only when they are anchored. Furthermore, there is some synchronization of somatic embryos development in which it is highly heterogeneous because of asynchronous development of embryos. All the differentiating cells and somatic embryos in a culture at the same development stage is desired prior to artificial or synthetic seeds technology or for studying the physiological, biochemical, and molecular changes underlying the process of somatic embryogenesis. The synchronization can be achieved by the physical separation of different embryogenic stages and physiologically by using growth regulators. For the physical separation, it is need to pass the heterogeneous suspension culture through nylon sheath of varying size or gradient centrifugation. Image analysis technique for counting somatic embryos could make way for computer-assisted sorting of embryos of different developmental stages. The application of ABA is necessary generally at the torpedo stage to prevent precocious germination. Alternatively, glycerol has also been used to achieve for the same result. Let's look into the physiological and biochemical aspect of somatic embryogenesis, especially the endogenous level of the phytohormones and their correlative effects. Once the embryogenic cluster are transferred to 2,4-D free medium, a rapid decline of endogenous 2,4-D metabolites and endogenous IAA levels after the globular stage, which means that 2,4-D is needed to induce the callus induction. Subsequently, it needs to be decreased by subculture it onto the media supplemented with activated charcoal or plant growth regulator free medium. Subsequently, the application of gibberellin and abscisic acid will be applied because less polar concentration of the gibberellin is found in the embryogenic callus as compared to non-embryogenic callus. Therefore, it was found that there is some changes in the metabolism of the amino acid, polyamines, and some enzymes. Theoretically, there are some molecular markers that used to determine the somatic embryogenesis. After the decline in the 2,4-D, somatic embryogenesis need to proceed with the maturation and the conversion of somatic embryos. It is because there is no accumulation of storage substances and the synthesis of ABA in somatic embryos as compared to zygotic embryos. The seedling generated from somatic embryogenesis is usually weak. Therefore, the maturation of the weak seedling developed from somatic embryogenesis can be promoted by using the ABA treatment or exposure to the high sucrose concentration and gradual desiccation. There are several abnormalities will be observed under the somatic embryogenesis. Abnormalities will be observed under the reduced humidity inside the culture vessel. The somatic embryos will exhibit the elongation for root and hypocotyl, but the regeneration does not occur. Another abnormality will be the oosin inhibitors could improve the conversion but increase the multiple shoots. It's commonly to see the standard somatic embryos that fail to germinate, or they will have precocious germination. Therefore, the concentration and the application of the plant growth regulator need to be optimized in order to have a successful somatic embryogenesis. As occurs with so many abnormalities, what are the advantages of somatic embryogenesis? First, it can be a large-scale production with less labor intensive. As compared with micropropagation, one shoot more likely to produce several shoots. Maximum, for example, will go to 10 shoots. However, there will be two stages whereby it need to be induced, the shoots first and then subculture to the rooting medium. So, it is time consuming and labor intensive. By using somatic embryogenesis, it will directly give you the regenerants with both shoot and root together. Furthermore, it can be used to produce the synthetic seeds through cryopreservation technique. Somatic embryogenesis is commonly used in the production of secondary metabolites by using suspension culture. It is a model to study physiological, biochemical, and molecular mechanisms for the onset of embryogenesis and development of embryos in higher plants. The disadvantage of using somatic embryogenesis is the response is tissue-specific. Different type of S-plant or genotype will give you the different result. There is a low frequency of embryo production in which you will also have incomplete embryo production. It may create unwanted genetic variation, which is the somal clonal variation as mentioned earlier. There is no true type as comparing to micropropagation. It has the inability to generate large number of normal plants that is free living plants. The plants regenerate through this method are generally weaker as compared to that of micropropagation. Here is a bonus of the somatic embryogenesis whereby there is some comparison between the somatic embryos and zygotic embryo. Zygotic embryo is the product of sexual reproduction, whereby the somatic embryo is known as the asexual reproduction. Zygotic embryo is formed by the fusion of gametes, whereas the somatic embryos is formed by the sporophytic or somatic cells. Zygotic embryos produce the seeds and contain the seed coat, whereas the somatic embryos only form the embryos and do not contain any seed coat. Zygotic embryo contains of the seed storage proteins and carbohydrates like endosperm, but for somatic embryo, it stores less amount of embryo-specific reserves. Therefore, it strongly relies on the supplementation of the nutrient media. A well-developed distinct suspensor is present in zygotic embryo, whereas a distinct suspensor is absent in somatic embryos. Zygotic embryos will have the vascular connection with the explant, whereas somatic embryos have no vascular connection with the culture explant. The plantlets raised by the zygotic embryo will not like mother plant, but they are healthy. However, for somatic embryos, the plantlets will similar to the mother plant, but they are weaker. Zygotic embryo do not show secondary embryogenesis and pluricotyledony. For somatic embryos, it lack of the dormant phase and often shows secondary embryogenesis and pluricotyledony. Zygotic embryo is known as the low rate of sexual propagation, whereas somatic embryogenesis gives a high rate of propagation. Before we end our video, let's look into the histological and anatomical aspect of somatic embryos. The cross-section of the torpedo can be examined. They will have two dense regions, which are the apical and basal parts. They show bipolar characteristics. There will be no vascular connection, which is the vascular system, xylem and phloem between the somatic embryo and maternal callus. Don't forget to subscribe, share and turn on notification. Please like us at the Facebook page and also follow us at the Instagram. If you have any inquiries and collaboration, please email to plantastic.mind@gmail.com. This is not a sponsored video, and I would like to share with every one of you the reference I used in this video. Thank you for watching and have a nice day.